opening the door wide to new possibilities, hope of real breakthroughs and understanding more big ideas than we could have ever imagined. These are the biggest grants we've ever been able to. We're going to come out of this with some breakthrough ideas.
Focused solely on neuroendocrine tumors. You are not alone. We are all with you on the board.
Our next speaker is Dr. Diane Reidy, medical oncologist at Memorial Sloan Kettering in the gastrointestinal oncology group, and heads up the neuroendocrine tumor program at that institution, and we're really honored to have her here today. Thank you.
Thank you. So I can't tell you how proud I am when I see a fellow who's now attending talk about neuroendocrine research. It makes me so excited. So that's number one. Second is that my two worlds are colliding. This is my first patient care experience, so to see my patients that I adore and care about so much actually in the room is a little bit different for me. So that's, I think, a good thing, but it's a change. So with that, I've been asked to talk about clinical trials. So obviously, this is a critical part of any disease. It's most important really in the rare cancers. You look at breast cancer, for example, there's 200,000 patients a year with breast cancer. We have thousands of patients in those trials that allow us to ask questions and provide really good answers. We don't have that in our disease. And in fact, our disease is so complicated because what's in a name? Our disease is many, many different types of diseases, right? And so because of that, we need these trials to help guide our management, but it can be tricky. But thankfully, in the last 10 years, which I hope to prove, we've really made incredible strides. So let's talk a little bit about clinical trials. I'm going to try to get to the clinical trials basics, the trials that are on the horizon, and what we can learn. And I should say my colleague and friend, Dr. Kuntz, provided some of these slides for me. So what's a clinical trial? If you go on to clinicaltrials.gov, they have this definition, which is clinical trials are research studies. They're experimental studies that we're trying to explore. It could be a medical strategy or a treatment or device, but we're trying to ask, first and foremost, is it safe, right? So first, do no harm. That's what we learn in medicine. And then the second thing is, once we figure out, is it safe? Then we say, is it effective? Is it efficacious? Does it help our patients do better and or live longer? So there's different types of clinical trials. So when you're out there and you're trying to explore the possibility of different clinical trials, you need to kind of know this jargon because it's really important to better understand what trials you may be interested in. So first you have preclinical studies, which you won't be seeing on the internet in terms of participating, because that's for the mouse. So it's mouse models, it's lab models, it's in vitro, it's, my patients come in and said, they cured cancer in this cell line. And I'm like, yeah, we did that a lot of times, over and over, but we're not cell lines and we're not mice. And God willing, one day we will cure these cancers, but right now, those preclinical models help us then go on to develop clinical studies. But these are just preclinical. So when you're reading about preclinical studies, you say, that's interesting. That's something that maybe they could do in the clinic, but we're not there yet. Then we have phase 1. So again, first do no harm always comes first. So the phase 1 is to say, is this a safe drug? And so right now, the vast majority of our trials do the following. I have a drug, that preclinical model told me it was gonna, cure cancer and cure neuroendocrine, and so we wanna test it. But we have no idea in the human, one, what the safe dose is to give. So we generally put 3 patients on, we give them, we'll say 5 milligrams, because we're making it up, and we watch them very carefully. And then if they do well, the next 3 go on at 10 milligrams, and then they do well, and then the next 3 go on at 15 milligrams. So, but for the vast majority of these trials, you're really not going up on your dose. So one can imagine you may be underdosed, you may be overdosed. And so what happens is, say you get to 15 milligrams and then you say, oops, somebody got a little sick, so we stop the trial. And then we add on a couple more patients and then we end up sometimes lowering it. So the phase 1s is really, is the drug safe? And if so, what is the dose required that won't be too toxic or too unsafe for the patient? Then we get that. So the phase 1 is now complete. And obviously, we're always hoping that in that study, the doses that we're providing is also helping the patient in terms of being efficacious, meaning that it's going to shrink the disease. But the purposes of the phase 1 trial is really to help us better define the safety of that drug. So now we get to the end of the phase 1 study and we say, okay, we got our dose, we know it's safe. Now I really want to test that 10 milligram dose that came out to be safe to make sure it can either shrink the tumor or keep it at bay and help our patients do better. That's the purposes of a phase 2 trial. So phase 2 is looking at efficacy. Everybody gets, with rare exception, the drug. There are randomized phase 2s that may not necessarily work that, but again, the question here is really how does the drug work? And then a little bit more on the side effects. Now we're saying with this dose and a larger number of patients, what are the side effects that we can see? And then phase 3 takes that drug from phase 2 with the same doses now and compares it to the standard of care. So we're taking this new drug and we're either randomizing it to placebo or randomizing it to the treatment that we have today. So those are the different types of phases when you're looking through clinicaltrials.gov or in other places to better understand where your trial of interest may be. And that's really important when you're talking to your clinician to better understand the trials that you're about to participate in. And neither of them are necessarily bad or good, it's just you need to know what they are. So a lot of times, again, people say, well, how do I know if I'm going to be able to be in this? And it's not that we as investigators don't want to put patients on these trials. Again, it goes back to that safety issue. If your liver enzymes are very high and the study is saying you're not eligible, it's probably because there's a potential for that drug to cause really bad liver injury. And so we can't put on patients that may be vulnerable and may have risks of potential harm. So the eligibility is generally, one, to make sure that the drug itself won't harm the patient that's participating. But then there's certain eligibility criteria based on the tumor type. I said earlier, neuroendocrine are a lot of different types. So sometimes there's well-differentiated, sometimes they're poorly differentiated. Those are different diseases. So when you're looking up the eligibility criteria, you want to know, does your tumor poorly differentiated or well-differentiated? And if so, is that aligned with the clinical trial that you're interested in doing? Many of these now are more site of origin. I'll talk about a couple of trials that we have that are focused on that. So is it just for pancreatic neuroendocrine tumors or is it just for lung neuroendocrine tumors? Knowing that can be also very important. Some of these studies do require or do allow, if you will, to have stable disease, but many of them require that your disease has to grow. And again, it comes back to safety. If you're doing well, maybe you don't need a trial. We don't want to put you at harm's way, at least not yet. So it's never trial or no trial. It's often timing of when these trials are best suited for you, which is really important. So if your doc says you don't need a trial, it doesn't mean that she's saying it that you never need it. It may just be today you're doing well and that there may not be trials available, but there's always an interest at least exploring, which I think is a great idea. And then how do these trials get run? So most times there's something we call an endpoint. So the investigator that runs the study has to say, we're looking at tumor shrinkage, we're looking at if the quality of life is better and their diarrhea is improved, we're looking at did the cancer treatment put the brakes on the disease, we call that progression-free survival. We're looking at all these objectives when we're running these trials to be very objective on does it work or not? And then ultimately, with the eye on the ball, most importantly, would be overall survival. And that last part is really important and tricky in our disease because, thank heavens, our patients can and often do live for years. So measuring overall survival can be hard because a lot of the patients eventually will go on these drugs, so it's hard to know in our disease with an overall survival endpoint. But that's something we always look at. So there are different types of clinical trials that are also offered, which are important. One is called an investigator-initiated study. So this is, for example, if I have an idea based on a preclinical model that we developed at Memorial, I will go to a pharmaceutical company. I say, this is a really good data. We put in what we call a letter of intent and ask them, would you be willing to support this trial based on our data? That's known as investigator-initiated. Then you have industry-sponsored trials where the idea came from industry, and now they're going throughout, the different communities and academic institutions to ask if, they would participate, and they're usually very solid ideas, again, but coming from preclinical in the pharmaceutical industry. And then you have cooperative groups where that's actually mostly government funding, which there's not a lot of, if you missed that memo, but they're supported by NIH, and we all put a lot of effort into this. And the studies are really important studies that we feel need to be done, but there may not be an interest in pharma to necessarily conduct those studies, which is why we perform them. And the funding there is federal. So why our clinical trials are important, I think I sort of alluded to this already. They're key research tools for advancing our medical knowledge. And they're really done to see, one, whether the new approach is safe and effective, and what treatments would be appropriate for which illnesses and why. Despite all of that, only 3% of patients with cancer enroll in clinical trials. So I think it's a conversation that really needs to be had. Really, it's the only way to really understand these treatments so that we can get better therapies in the future. So what do we have? So the good news is that for neuroendocrine tumors, we're making other cancers look bad because we really have had a lot of approvals for this rare disease, unlike many other cancers that I take care of, for example, including pancreas, colon, gastric. We've had a number of FDA-approved drugs. We had streptozotocin in 1982, which was terribly toxic. And then, as you've heard from Dr. Wolin, we've had several other therapies in the last 5 to 10 years that were recently approved. And that's the good news. So the not so good news is sometimes this is a mess, right? So how do we decide for any one patient what treatment options should be provided? And it's almost impossible when patients say, what about this, and what about this, and what about this? And we try as best as we can as clinicians to explain it to you. But there's so many different, again, types of neuroendocrine, and thankfully so many different types of treatment options that the art of medicine comes in here a lot. So I'm not asking you to, I'm not going to go through this, but again, there's a lot of complexity here, and sometimes it's great to have, patient support and colleagues, but it can get a little tricky sometimes because if you compare notes, you're like, well, why did you get that? I got this. And it's in part because of all these complexities of the different types of diseases that we're treating at any one time. So what types of trials do we have? So the famous gallium, I think many people in this room probably know about the gallium-68. Gallium-68 is what we call a non-therapeutic trial. It won't make us do better. It won't make us live longer, at least not yet. It's a test. It's a PET scan. And the idea here is that for all of you who know about Octrio scans or have family members or if you're patients on Octrio scans, it's a hard test. It's very expensive. It is very timely. Patients get the injection of the radiation. Then they have to wait, then they have to come back the next day, and then they get all these pictures. Sometimes they have to come back the third day. And it's a lot of radiation as well. And so the gallium-68 is a type of PET scan that is much more sensitive than the traditional Octrio scan. And it allows us to see if the tumor has this receptor on the outside of the cell. And if it does, the tumors, when you inject this little bit of radiation with a protein, the radiation goes to the tumors, and then they light up like a Christmas tree. So having the tumors light up like a Christmas tree like that tells us a couple of things. One, the potential for using therapies like somatostatin analogs and PRRT, which we'll talk about. And it also allows us to get the extent of the disease. Could there be some sort of small little bone lesions? So we did in several institutions, including Dr. Wolin here, had this on study with a gallium-68 DOTA-TOC. And then just recently in June, the so-called NET spot test was approved, which was great in June. The problem is there's been a lot of growing pains and hiccups in getting these and generators and that kind of thing. So many institutions still don't have this test. I think it's a good baseline test to see, does your tumor light up or not light up? But it's a lot of radiation too. So you don't want to be repeating these without good reason over periods of time. So it's an important study that was approved, this NET spot test. It is definitely going to be helpful to our community, but where and how often this test comes in is still sort of unknown in that way. And many of us are working with nuclear medicine societies to better define those sort of indications and reasons for these tests. So I've been asked to talk about what trials are going on. So at the expense of doing laundry lists, I'm going to try to go through some of them and the importance, but very briefly as an overview. And after the talk, certainly if there's any questions, I'd be more than willing to talk to anyone. So this is where we're going to focus first on the pancreatic neuroendocrine trial. So this is a study, again, from the cooperative group, so NCI funded, of the combination of temozolomide capecitabine or CAPTEM, that people may know about. Very small study, only very few patients, but a dramatic response in pancreatic neuroendocrine tumors. But no one really ever looked at the role of 2 drugs versus 1. And so that's what this study is doing. Like, do you even need the capecitabine? You could say, well, the study is so good, why do you even have to do it? But again, if we can get away with one drug instead of two, then maybe we can use the capecitabine next or after that, and maybe patients can live better and longer. So it's a really important study, but you can imagine why this didn't really get that much excitement in pharma, but NCI does. So this is an example of why we need to have team efforts for these types of studies. Then there's an interesting study of called Reminet, which is in pancreatic neuroendocrine tumors. So essentially they have advanced either small duodenal or pancreatic neuroendocrine tumors. They get chemotherapy for a couple of months, 3 to 6 months, and then they go on a maintenance therapy with lanreotide. And they randomize them to get this maintenance somatostatin analog versus placebo to see who does better by being on some maintenance therapy. And again, you could say, well, I don't want placebo, but this is one of these things that, thank heavens, our patients will live for a really long time. So if we find in this study that maintenance therapy didn't make a difference in their overall survival, getting away with avoiding the shot in the bottom once a month could be a very exciting thing for our patients in terms of quality of life. So these are questions, again, that are really important to do. Sector is also a really important one because none of us know, we don't have great data to tell us which therapy should be done in which sequence. Most of us start with somatostatin analogs, meaning octreotide or lanreotide first. We call them our first string players because they're so well tolerated. But what if in pancreatic neuroendocrine tumors, after the somatostatin analogs, we did everolimus, the mTOR inhibitor that Dr. Wallen talked about? Or what if right after that we use cytotoxic therapy, such as streptozocin? Does it matter if you start with a chemo or if you start with an mTOR inhibitor or not? And so we don't know that answer, and so this is a study which we call sequencing, where we're trying to define, if you use targeted therapy first or cytotoxic therapy first, which one wins, so it's a really important study to do. We heard a little bit about pazopanib. So this is another sort of inhibiting the blood vessels. This is for non-pancreatic neuroendocrine tumors. This is a study that actually is accrued? Fully accrued, right? We just finished, which is great. So comparing in the so-called carcinoid or non-pancreatic neuroendocrine tumors and combining, randomizing it to placebo. And then we have a study in high grade. And for our patients with these poorly differentiated or high grade neuroendocrine cancers, it's very frustrating. One, because they're really rare, but two, we really don't have trials for them. And so we're working very hard to do these types of studies. And this is a study that randomizes 2 different types of cytotoxic therapies, platinum etoposide versus CAPE-10. And then the last one is the SPINET, which I'm going to talk a little bit about. And this is, in somatostatin analogs, we now have 2 studies that show in GI neuroendocrine tumors they work, but the poor lung nets have been really sort of avoided, and they really can't and shouldn't be avoided. And the issue there is that for so long, people were treating lung nets like what we call small cell neuroendocrine carcinoma, which is a completely different disease. And so we really don't know, do somatostatin analogs work in lung net? And if so, how well? And so this study is actually asking that question. In lung nets, do somatostatin analogs work when you compare it to placebo? It's a 2-to-1 randomization, so you have a very good chance of getting the drug. And if it grew a little bit, then you can get the drug afterwards, because it's not FDA approved for lung nets. So we're trying desperately to get this study done so that we can have the answers for our patients with lung net. And then we heard about Netorone, which was what we consider a home run in the world of cancer research in terms of really improving the progression-free survival. The good news, again, is it definitely improved the progression-free survival. The concern with this drug is, again, it's a little bit of radiation tagged to a protein. So that radiation could potentially go places that we don't want it to go, which is namely the kidneys, although now the kidneys are pretty safe with the therapies that we use to protect it. But the longer-term potential risk is the bone marrow, the red cells, the white cells, and the platelets, which rarely but seriously can be irreversible, and then very rarely but seriously could lead to leukemia. So we do have some data right now that suggests at least early on it's safe in this prospective study, but we need longer term to really define the true toxicities, because again, thank heavens our patients can do very well for very long, but if we, God forbid, have a bad toxicity like leukemia, we're obviously in trouble. So is this true, Ed? Are you open? Okay, beautiful. So Dr. Wallen was first out, and he has this open. Now, this trial was complete. It was, again, a home run. We now have an expanded access program for patients with small bowel neuroendocrine to participate in this study. It's open now in Monty. We have it going to our IRB next week, so we will also be open within the next couple of months. This is in patients with progressive small bowel neuroendocrine tumors, but a great study to consider, and you should certainly, most certainly, talk to your clinician about it as an option if open in the future. I talked about this already. This was the lung one that I thought was really important. And then the last one I wanted to just talk about in terms of therapeutic studies is sort of a second generation somatostatin or PRRT. So we call this version 2.0. So the PRRT in the NETRF study was a little bit of radiation linked to octreotate. And this is a different protein, if you will, that we're linking it to. That's what we call a somatostatin antagonist. And in the preclinical studies that we discussed earlier, we found that at least in the mouse models, there was a lot more uptake in the tumor and thereby a lot less in places like bone marrow and in the kidneys. So this is the study. This is a phase 1 study. So again, we're really looking at safety and hoping for efficacy. But essentially, we have 19 of 20 patients. This study is on hold now because we're just essentially at accrual. But we're really excited to see those results moving forward. And this is just an example of what happens when we say your tumors light up like a Christmas tree. So this was our first patient study. She's 19 years old and completely asymptomatic with all that disease. And that's what can happen in our tumors. But this is also the uptake with the gallium JR11 that she received. So really could potentially have a tremendous difference for many of our patients that have very avid disease there. And this is the next 3 patients that were enrolled. Again, patients that had very avid disease, and we're hoping, with our fingers crossed, that the radiation-induced PRRT will shrink these tumors. So then I'd like to end with sort of bringing it back to the lab. So a big part of all of our trials is what we call correlative studies as well. So every trial that we do, it's not enough to, I mean, we most certainly are always focused on the patient to make sure if we can that these treatments will help the patient, but we have to also learn as much as we can during that. And so many of our studies have gene sequencing and other correlative studies and immunotherapy markers and other things to help us better understand what's happening when the patient gets the drug. So we're very privileged at Memorial to have something called next-generation sequencing. The good news is in many patients, we can actually define the drivers of the disease based on the genes that we identify, and particularly the genes that are damaged or the mutations. The problem is in our disease, we don't have a lot of mutations. And it's probably one of the reasons why our tumors grow slower than other tumors, so that's the good news. But the not so good news is it's harder to use this type of therapy, or I should say use these types of tools in most neuroendocrine tumors. But in pancreatic neuroendocrine tumors, it turns out that they do have a lot of mutations. So that's what we focused our efforts on. In LungNet, there's also mutations that we're doing. But I'm going to share the pancreatic neuroendocrine tumor data because for small bowel nets, unfortunately, we just didn't find a lot of gene drivers. It may be more what we call RNA or protein level or epigenetic stuff, but it's not usually a driving mutation. But this is a study that we had. This is now up to 80 patients. And what we found, very briefly, is that when you do all these genetic sequencing, it's important, one, because certain genes that you may find that are damaged could help us prognostically. What I mean by that is if you have this gene, there's a good chance that this cancer could be very aggressive. So that's helpful to share with a patient in case we find it. But also there's a potential that we may find a gene that we could target in the context of a clinical trial. And we call those basket studies. So what we find is that if you have a gene that's altered, there are therapies in these basket studies that actually can target those genes. So we conducted this study, and in fact, we did find genes that could be targeted. So this is a patient with what we call a TSC2 mutation, and this is along the mTOR pathway, again, that we heard about from Dr. Wallen. And lo and behold, this patient did have a nice treatment response. Now, whether it was from the mutation or not is unknown, but interestingly, after the patient's disease grew, we biopsied it, and then there were new mutations that we found along the mTOR pathway, which is pretty cool. So we actually can define, in some patients, why, or we think, maybe, why the tumor stopped responding to the drugs. This is another patient that had a BRAF V600 mutation that we have drugs to target, pancreatic neuroendocrine tumor that had the tumors shrink, and she sustained a response for about 9 months before the disease unfortunately progressed. So the last part was we were finding a lot of mutations along this pathway called CDK4/6. And so as a result, we now have a study, and the mutations were really found in pancreatic and gastric and duodenal nets. And so as a result of that, we had an investigator-initiated study. If you remember, that's an idea we have that we take to pharmaceutical companies. And through Memorial, MD Anderson, and Dana-Farber, we're just about to start this. This was actually just FDA approved last week for us. and we have a combination of everolimus plus the CDK4/6 inhibitor for patients to participate in. So the future directors, again, combining the therapeutic, the non-therapeutic, and the correlative studies. And then we're working a lot on mouse models, actually through Dr. Labudi, who unfortunately is not here. He's helping us a lot on these mouse models with my colleague, Dr. Anj. So lastly, but certainly not least, how do you learn about these clinical trials? You go to clinicaltrials.gov. You put in neuroendocrine tumors. Again, you can search by phase, so you could put either phase 2 or phase 3, or if you're interested in phase 1s, I would say phase 1s are always potentially a good idea to participate in. It's a little bit challenging to be a phase 1 if you're living very far away. So, you know, phase 1 that is at Memorial when you're living across the street is much more doable, if you will. But phase 2s and 3s are trials that you could more easily participate in at a distance. And then lastly, academic institutions have their websites. I put up Montefiore and Memorial here, but certainly Mount Sinai is a very big program in neuroendocrine as well. So going to the individual academic institutions can be very helpful. So the take-home points is that we certainly have more tools. Our patients and our treatment characteristics, again, through these different treatment algorithms can be really helpful. And these smart trial designs really are imperative to ensure that we move our field forward and provide the necessary care for our patients. So thank you very much.